Hey guys, so we are going to rock out the last two sections in this video number 4 for chapter 20. So we are going to talk about ELISAs, EIAs, and fluorescence in this video. We're going to talk about the last bit of content for chapter 20. So yay, last one. Okay, so how do ELISAs work? They're going to be similar to a Western blot, kind of, sort of, in that we are going to be looking for the antigens. They're going to attach onto an antibody, and it's going to fluoresce. So that antibody is going to have either a fluorophore, which is going to be the section 5 video, or it's going to have a chromatophore, where the chromatophore will change color depending on what's going on. So in this case, with the enzyme immunoassays, the EIAs, they are going to detect the presence of the antigen, and we are going to be able to see them. This is a picture of a direct one. Direct ones are quick and dirty. They're quick and easy for direct ELISAs. They have an antigen, and they're going to have the antibody with the enzyme on it attach on next. So it's just that's the next step. It is directly doing this ELISA with the antibody and the enzyme on it. Then we add the substrate, and boom, you're going to see a color change, or you see nothing because it's a negative. So this is a direct ELISA. Other things that we talk about in this section are going to be immunohistochemistry. So it's got histo in the name, and histo means tissues. So with the immunohistochemistry, we are looking for cells involved in the tissues. And in this example, we have cells of, what is this? Bone marrow. It is bone marrow. And say we needed to know what cells are in this tissue, how many they have in here. So I think the textbook was talking about if you were looking for AIDS patients, for example. We said that AIDS patients, if they are HIV positive, HIV is going to attack your helper T cells, and they were super important. How many helper T cells a person has determines if they are progressing from HIV into AIDS. So you may need to actually count those cells. So this is a way for you to do that. With the immunohistochemistry, we can attach a marker onto these things so we can see this tissue, stain it, and pick them out of here. We can also use immunocytochemistry, and this is where the cells are messed with first. So instead of just staining the cells and boom, we can see, the helper Ts. With the immunocytochemistry, they mess with the cell membrane on the outside first. So they etch it with alcohol, and that way those antibodies are allowed to pass through the cell membrane when you mess with it that way with immunocytochemistry. So we can see the components and the structures and all of that when you mess with the cell membrane. Things can get through. So these are immunohistochemistry and immunocytochemistry. Histo, staining it and being able to see the individual cells within the tissue. And cyto is when you mess with the cells and their cell membrane, and you can mess with the permeability, basically, of those guys. Let's move on. So we saw the direct ELISA, antigen, and then here is going to be our antibody, and it's the very first one, the antibody and the enzyme complex on there, and then we add the substrate and you see a reaction or you don't. That is the direct ELISA. Not to be confused with the sandwich ELISA. So on the direct ELISA, antigen's on the bottom, and the antibody enzyme complex is attached on directly to the antigen that's on the bottom. Now, for the sandwich type, the process is sort of similar to gram staining. When you gram stain, right? You fix your smear on there with your bacteria, and then you put your stain. You let your stain sit, then you wash it off. Then you do a counter stain, let it sit, wash it off. So it's kind of the similar steps here on the sandwich ELISA. Here, we have fixed our stuff with our primary antibody. That's first. Then we're going to rinse. And then we're going to add a blocking agent, step number 2. And then we're going to rinse. And then we're going to do an antigen. So hence, this is like the peanut butter and your peanut butter jelly sandwich is your antigen right here. And then we're going to rinse. So if those antigens are the right antigens for the antibodies that we have on our locator on the bottom of our microtiter pipette plate, on microtiter plate. If those antigens are correct for those antibodies, they stick. If they're not going to stick because, you know, antibodies are super specific. If they're not the right antigens, they wash away when we rinse. So moving on to step 4, they get washed away. Step 5, now we're going to the other slice of bread on our sandwich. Now is our secondary antibodies, and they attach on. So our antigens are sandwiched in between the first primary antibody and your second antibody. And that second antibody is going to have the enzyme complex on it. Then we're going to rinse again, because we always rinse. And now step 7, substrate. We add the substrate, and we don't, there's nothing going on here because we don't have the antigen, which didn't bind to the second antibody and the enzyme, and we're not going to see anything over here. But over here, we did have the antigen that stuck to the primary antibody that is sandwiched in between the secondary antibody and the enzyme. And when we add the substrate, we do see the reaction. So this is a sandwich ELISA, different from a direct ELISA that just had the antigen and the antibody enzyme complex added directly to it. Sandwich ELISA. Indirect, what are we looking for? With our indirect, we are going to see what type. So we're going to start, instead of starting with antibodies like we started with on our sandwich, for the indirect, we're going to start with the antigen. So kind of similar to the direct, but it requires the 2 antibodies. So we start with our antigen, and we are going to see, does this sample contain the primary antibodies? Does it have them? That's what we're looking for. So if the subject does have the primary antibodies, then they will stick to the antigen. If the subject has the incorrect antibodies, those won't stick to the antigens because, again, specificity. They will wash away when they get rinsed. So the primary antibodies, they stick here. And then we're going to have the secondary antibody stick to the primary antibody. So it's not a sandwich because the antigen was first. It's not the peanut butter in the middle of the sandwich. This is an indirect ELISA. So it has antigen, primary antibodies. That's what we were looking for in our patient. Does the sample have it? And then here's our secondary antibodies with the enzyme, rinse. And then going to add our substrate, and we get a reaction going on over here. So this is an indirect. So we've talked about direct ELISAs. We've talked about sandwich ELISAs with the antigens in the middle, like peanut butter and jelly. And an indirect ELISA where the antigen isn't in the middle, it's the primary antibody. That's what we're looking for our indirect ELISAs. Checking in with our case study with that person, the guy who were switching out syringes, switching out needles for things. And so they wanted to contact 1,300 patients that they determined that this guy had contact with. So they've gotten in touch with those 1,300 patients. They are going to see and test them for HIV. Oh, excuse me. Because the sooner you test, the sooner you can know, and the sooner you can get treatment. So this is literally life-saving. The faster they work, the better, to try and determine, did this person infect anybody else? So, variety screening tests, going to use the indirect ELISA because we're looking for those antibodies. Do they have antibodies to HIV? We said that, do they have it out here where they're talking about how long it's going to take? Okay, it's just explaining to you what the process is with the indirect ELISA, that we are looking for anti-HIV antibodies. If they have HIV, if they're HIV positive, they will have anti-HIV antibodies that will bind to that antigen in the indirect ELISA that we just talked about. All right, a lateral flow test. Some of us are familiar with lateral flow tests. Some of us may have actually used a lateral flow test either in your work or personally. Lateral flow tests. So with these, the reagent is going to be dried up and stuck into the strip because these are, lateral flow tests are really quick and easy. The reagents are in the strip, and the substance that you are testing, urine, whatever, that liquid will come across the strip. It will, you know, come across capillary action. It's going to draw it across the strip, and it will set off a chain of events. So with the lateral flow, we're going to say that this is a pregnancy test. It is a pregnancy test because it is looking for human chorionic gonadotropin, HCG. HCG is the pregnancy hormone. This is a pregnancy test strip. So mama is going to urinate on the test strip, and then it is going to go across these HCG specific antibodies. And here's where they're going to mix first. So the urine is going to capture these HCG antibodies, and they're going to bring it across. So now it's going to hit the test line, and the test line is going to have the other antibodies that will attach onto those. So they are going to have our HCG first antibodies, these little green guys, and they will capture. When they capture this, they are going to coagulate, and it can either be, let's see, antibody, colloidal, gold coagulates, or the gold is going to give you the red color, or the latex beads are going to give you the blue color. But we're going to get a color in this situation because the person did have the HCG that's captured by the antibodies, and then those antibodies stick onto the HCG first antibodies. So this is our lateral flow going on. So we got a sandwich going on. And then as it continues on down, those guys are then going to also set off the control line that we're looking for to see do they have the HCG antibodies attached onto the HCG. And if they do, it sets off the control line too. So this is your lateral flow. Urine comes in. If they have, do they have the antigen? In this case, the antigen is HCG. It will bind here with this HCG antibody that will attach to it. Then it will become a sandwich ELISA over here, and it will change color. We are going to have gold conjugates. Here we go. Oh gosh, cannot speak today. We're going to have gold conjugates, and it's going to change the color. Gold is going to give you red. Latex beads are going to give you blue color. So we're going to have this line. This person does have HCG in their urine. So we have the test positive line that's going to color. And then our control line, just to make sure that it works, is going to go off because the urine is going to cause all of these guys to flow here and eventually flow here. And these are what sets off your control line. So are these guys able to make it over here? And they should, no matter what, and that will set off your control line. If somebody urinates and there's no HCG antigens, you are going to have these guys go across, but your sandwich ELISA is not happening because you don't have an antigen to set it off and to capture those. So you skip your test line. This is going to be negative, but your control line still gets set off. So this is your pregnancy test, looking for HCG. Will this go off if you are testing yourself, 24 hours after intercourse? No, it won't. You need it to build up in your system to be able to set this off. So this is your lateral flow test. I have my own personal story regarding this. With my oldest, I was in grad school, and my background is cats, is feral cats. That was my master's. I was working on feral cats. And so we would trap the cats, neuter the cats, vaccinate the cats, test them for FELV and FIV, and then we would release them back as part of this trap, neuter, return program. So I used FELV test kits. And with the FELV test kits, there's 2 lines. So I had this embedded in my brain. There has to be 2 lines for it to test positive. Well, apparently the pregnancy test that I took the first time only had 1 line. And I don't know, like, if the control was just not shown or what it was, but there was only 1 line. So I just assumed that I wasn't pregnant. When in fact, the way that test was, I was reading it incorrectly. It was user error on my part, and it really was pregnant. So it took another 2 weeks for me to be like, and it took a totally different pregnancy test that was a little bit, easier to read. And then it was like, oh, that's it, yeah. So now you know how the lateral test kits work, and that hopefully you'll be able to read and, you know, be a little bit smarter than I was way back in the day, way before I ever took microbiology or knew anything about this. There should be a control line and there should be a test line, and you should see both of them. And you know how it works and what it's indicating. So now you know. Now you know. Okay, so those are lateral flows, and we talked about ELISAs, direct ELISAs, sandwich ELISAs, and indirect ELISAs. And we also talked about the immunostaining. The last section that we need to talk about, of course, before we get there, case study update. Okay, so we said that they were going to test everyone for HIV. We are looking for an indirect ELISA because we want to know not if they have the antigens, we want to know, do they have the antibodies against it? Do they have the anti-HIV antibodies? So we have to wait, right? Because we said that your body producing the antibodies against an infection takes at least 3 weeks. Same for flu shot. If you have a relative that tests positive for flu and you run out and get a flu shot the next day, the body does not have the antibodies against it. It takes 3 weeks. So with all of these people, those 1,300 people that they need to test from the hospital, they got to wait 3 weeks. They're going to get in touch with them. They're getting all their ducks in a row. And they're going to use an indirect ELISA. It can yield false negatives if they go too early. So they're going to wait the 3 weeks. They can also have false positives. So just in case these happen, they're going to have counselors for anybody who tests positive ready and waiting. And then if they get a positive test, again, to rule out if it's a false positive, we're going to do a secondary test. So we're going to have a confirmation test. It's going to be a Western blot looking for those HIV proteins. So we saw a picture of those Western blots earlier, a couple slides back in the last video, I want to say. And you could see, so in a Western blot, you're doing the electrophoresis, you're doing the page to separate out the proteins, and then you're looking for the individual ones. Do they have the HIV proteins? Do they have HIV protein 1? Do they have HIV protein 2? Do they have any HIV proteins at all? So we're going to use a Western blot to confirm any positives coming from these indirect ELISAs. And then, here we go, a positive Western blot would confirm HIV infection. A negative would say that it was a false positive. Okay, so they've done the 1,300 patients, all with indirect ELISAs. We got some positives. Now we're going to do the Western blots to see if there's any false positives or if those, you know, in the second test are positives. Okay, so some of the Western blots are indeterminate results. So they cannot confirm or invalidate. They don't know. That happened with my husband in COVID this summer. He had every box, every symptom, every sign, all the things on the CDC website. He had every single one of those. We sent him in to get a COVID test. He gets a COVID test and it comes back indeterminate. We don't know. So we're just assuming he had it. They said to assume he had it, but we didn't have the proof in our hands to say that he had it. So it was never registered in the system. But sometimes you get indeterminate results. So if there is an indeterminate Western blot, then we'll need to do test number 3. We're going to do a third test. Okay, so the rate of indeterminates can be 10 to 49%. Holy cow. Okay, so then they are going to do another test. They're going to do another test. And I don't think they tell us what that other test is, but they will later on, and we'll get to that. Fluorescent antibody. So before those guys, with our lateral flows, they were going to color change. You were either going to see a red one, or you might see a blue one show up, depending on if it's gold or latex beads on those guys. With the fluorescence, it's going to fluoresce. It is going to glow a brighter wavelength light with fluorescent that you can pick up with equipment with our fluorescent antibodies. So if somebody had syphilis or Legionella or something like that, with our Legionella, some of these guys you can't culture these, you can't grow these and perform tests on them later. You have to use the patient's sample. and test from that. So you have to use those live, bacteria from the patient sample. And for that, you would need to do a direct fluorescent antibody technique. And so those antibodies will attach directly onto the bacteria, onto the cells, and then you can look with those to see if those fluorescent labeled antibodies attach onto, and then look through a fluorescent microscope and detect if those are present or not. If you're not seeing them fluoresce, if you're not seeing anything, then they don't have that specific bacteria that you're looking for. So those are direct, kind of like a direct ELISA. It attaches directly onto the antigens, but instead of having a color change where it's going to use the gold flecks or the blue latex beads and color change that way, here it fluoresces instead. So if you have the antigen, those antibodies have a fluorophore on them and they will fluoresce. You can pick it up with a microscope. Indirect, just like all of our other indirect ones, do they have the antibodies against the antigen with indirect? So indirect is going to have the pre-prepared antigen attached onto the surface. If the patient's serum does have the antibodies, those are going to attach. And then you're going to have your fluorophore coated antibody that will fluoresce under the microscope. So again, these are really helpful on bacteria that don't culture well and that you need to use a live one for these guys like Legionella. Or if you think a patient has been infected with something and they've got the antibodies against it and you want to test their serum to see if they've got the antibodies against it. Maybe maybe they had it, but they don't have it right now and you need to know if they've got the antibodies that are still in their system. You do use an indirect fluorescent antibody. We mentioned, was it last chapter? Was it chapter 19? It might have been. I can't remember her name, but she was having issues and they tested her and it turned out that she had lupus. Well, one of those tests when they determined that she was having hemolytic anemia, that her body was attacking its own blood cells, was the fact that they were looking to see if she had anti-nuclear antibodies, an ANA test, and that is going to use fluorescence around these guys. If you have it, it will fluoresce. If you don't have them, then it won't. And so previously, I want to say it was chapter 19, that patient had lupus, and this was one of the tests that they used, and hers would have fluoresced like this one does. Okay, with the fluorescence, how do you pick it up? The fluorescent microscope can pick it up. You can also not only detect it just looking through it with fluorescent microscope, but technology is super cool. We can actually count the number of cells. That's important for different reasons that you might need to know. How bad is this infection? What, is it just one cell or two, or is there a lot? What's going on? How many are there? So with flow cytometry, you got your cell samples. We have a labeled cell that is going to fluoresce. It is going to come through. It's going to get hit by the laser beam, and that is going to be detected up here. The laser beam does not go all the way through. It's going to be stopped right here, but it's going to cause it to scatter. The fluorescent light is going to scatter off. We will be able to pick it up and detect it with the computer. So we can count how many cells have this fluorescence going on out of our sample of, X number of cells. Another super cool thing is when we detect that we have a fluorescent cell in this situation, we can count it, which I think is super cool. So if you have a fluorescent cell, it fluoresces at that time, then it can put in a charge to the water surrounding that, to the serum surrounding that area of that cell, and then it will shunt that into a different direction, and then the other cells will go into this direction, so we can count how many are fluorescent. And in this case, here's our cell count. These are the fluorescent ones that express the CD8, and these are the ones that did not express CD8. You can count them and separate them. Super cool. Okay, so resolution. The hospital tested all 1,300 patients after 3 weeks after they had been exposed to that employee. There were some false positives from those indirect ELISAs. And so let's see, we had eight 8 tested positive using the ELISA test. 5 of them were invalidated by negative Western blots. Okay, so the Western blot was our test number 2. We had 8 positives and 5 did not have the proteins found in the Western blot, so we throw them out. So now we're down to 3 patients that might potentially have gotten HIV due to the circumstances happening at this hospital. Okay, 5 were invalidated by the negative Western blot test. 1 Western blot came back positive. Okay, so we definitely need to keep going with that one. That confirmed that that patient did contract HIV. 2 of the Western blots were indeterminate. We can't tell on this Western blot what's going on with them. So out of the we don't knows, we have to go to a 3rd test, which is PCR. And they're going to see does HIV's genetic material, is that present using the PCR? We will look directly for HIV's nuclear material with a PCR. And both of those tested negative. Okay, so out of our 8 indirect ELISA tests, 5 of them got thrown out when we did the Western blot. 3 of the Western blots were iffy. 1 of them was definitely positive. So we know 1 patient out of there, out of the 8, is positive. 2 we weren't sure about, so we ran a 3rd test for PCR to look directly for HIV genome, and they did not find any, so they were negative. So 1 person out of the 1,300 patients since they started testing was determined to be HIV positive. And it cannot prove or disprove that it was due to these syringes, but they're not going to accept liability, but they are going to cover it. So the insurance is fully covering the patient's treatment, which began immediately because, again, the sooner you treat this, the better off the patient will be, and they will need this treatment for a lifetime because there's no cure right now at this moment for HIV, but we do have preventative drugs to prevent it from progressing. Yeah, talking about the drugs that we use to stop it, slow it down. All the different tests that we talked about in this video, that is it. So chapter 20 was a lot of tests. It was a lot of information. If you have questions, please shoot me an email. Have a great one.